Bismillahirrahmanirrahim. So our today's first topic is beta-oxidation of polyunsaturated fatty acids in mitochondria. If an unsaturated fatty acid has more than one double bond of cis type or at wrong places, for example linoleic acid. In linoleic acid there are two double bonds. First double bond is at carbon number 9 and the second is at carbon number 12, and linoleic acid is 18 carbon compound. Then the complex reactions are needed for its beta oxidation. Now we will see its steps. First step is after 3 cycles of beta oxidation, 3 molecules of acetyl-CoA are released, and the linolyl CoA, which is the CoA derivative of linoleic acid, is shortened to 12 carbon fatty acyl CoA having cis double bond at the position of carbon number 3 and carbon number 6, जो कि हमारे पास पहले कार्बन नंबर 9 और 12 पे थे, अब हमारे पास 3 और 6 पे हो जाएंगे. So it becomes cis 3 cis double bond at 3 or cis double bond at 6 position, 12 कार्बन फैटी एसाइल CoA, और इसका स्ट्रक्चर हमारे पास ये आ जाएगा जो कि बुक में भी गिवन है. एंड इन द सेकेंड स्टेप द एंजाइम present at double bond at carbon number 3 and 2, which is enoyl CoA isomerase, converts the double bond present at carbon number 3 position to carbon number 2 position पे ट्रांस में कर देगा, सिस को ट्रांस में कर देगा, जबकि जो कार्बन नंबर 6 पे डबल बॉन्ड था सिस टाइप का वो अनचेंज्ड रहेगा. एंड द स्ट्रक्चर इज गिवन. Then a new cycle continues which releases 1 acetyl CoA and 12 carbon fatty acyl CoA is shortened to 10 carbon. Cis double bond at carbon number 4 position and trans double bond at carbon number 2 position. The structure is given. In the fourth step, the enzyme 2,4-dianyl reductase adds 2 hydrogen atoms, removing double bond at carbon number 4 position, which is cis, and trans double bond at carbon number 2 position becomes trans double bond at 3. मतलब जो 2 पे डबल बॉन्ड था वो 3 पे शिफ्ट हो जाएगा. The structure is given. Then the enzyme enoyl CoA isomerase shifts trans bond double bond at carbon number 3 position to trans double bond at carbon number 2 position. The structure is given. Then 4 cycles take place smoothly and the beta oxidation completes. So that was the beta oxidation of fatty acids in mitochondria. Now we will discuss beta oxidation of fatty acids in peroxisomes, जो कि स्पेशली हमारे पास लॉन्ग चेन फैटी एसिड्स की होती है. फॉर एग्जांपल अ फैटी एसिड containing 26 कार्बन और मोर एटसेट्रा. इन बीटा ऑक्सीडेशन इन peroxisomes during FAD dependent oxidation of fatty acyl CoA, electrons are released and they are passed to the molecular oxygen to form hydrogen peroxide. Then the enzyme catalase present in peroxisomes converts the hydrogen peroxide into water and oxygen, because peroxisomes do not have respiratory chain. Next is alpha-oxidation of branched chain fatty acids in peroxisomes. Alpha-oxidation metabolizes branched chain fatty acids. This alpha-oxidation is important in utilization of phytanic acid. The structure of phytanic acid is given, and this is also called as 3,7,11,15 tetramethyl hexadeconic acid. Beta oxidation cannot occur because beta carbon is blocked by methyl group. So in alpha oxidation, the first step is phytanic acid is converted to its CoA derivative, that is phytonyl-CoA. In the second step, hydroxylase enzymes introduces a hydroxyl group at alpha carbon. Then the new compound is alpha hydroxyl phytonyl-CoA. Then two enzymes, alpha hydroxy phytonyl-CoA lyase and aldehyde dehydrogenase, remove the original alpha carbon, resulting in a fatty acid with one less carbon, that is pristanic acid, whose structure is given below. Now, successive beta carbon becomes available for beta oxidation, as we can see. In the branching sites, the product of beta oxidation will be propionyl-CoA and not acetyl-CoA, as this is clear कि ये अल्फा ऑक्सीडेशन जो है ब्रांच्ड चेन पे है, तो हमारे पास प्रोपियोनाइल-CoA ही बन सकता है, एसिटाइल-CoA नहीं बन सकता. And then propionyl-CoA is converted into succinyl-CoA, and succinyl-CoA is metabolized by citric acid cycle. Phytanic acid is ingested in diet containing food of animal origin, for example cow's milk. In certain individuals, alpha oxidation does not take place due to hereditary defect, which is autosomal recessive in character. The accumulation of phytanic acid in various tissues produces phytanic acid storage disease, which is known as Refsum's disease, and the symptoms of Refsum's disease are failing night vision, retinitis pigmentosa, deafness, peripheral neuropathy, and cerebellar defects. Alpha oxidation is also used to oxidize 2-hydroxy fatty acids, which are found as components of brain lipids. Thank you.